I'll be discussing about drug design and application of biovia in life sciences. So the term biovia, which represent biovia discovery studio. Probably you people are aware of Schrodinger as well as discovery studio. Okay, so here we'll be discussing about biovia discovery studio. It's a commercial software and how we are using bioinformatics with this software. And specifically, I'm going to talk about homology modeling. So after homology modeling, so I'll be discussing what are all the techniques are available in drug designing approaches and all. So before we start with the presentation, I'll give a small introduction about my company. So we, Altem Technologies, we have started in the year 2010, and basically we are from a 3D printer and scanning company. And recently we have started BioVia Life Sciences, and which comes under Dassault system. And we are into life sciences research, as well as we have started the 3D bioprinting right now. And this is all about my company. And coming to bioinformatics and cheminformatics, hope from this panel members, you will be aware of bioinformatics as well as the cheminformatics terms. It's nothing but bioinformatics is like biology in computer science and chemistry in computer science. So that's all. Now how we will be using bioinformatics as well as cheminformatics approaches in drug design as well as a target discovery approach. So say for example, there is something called biological lead identification. Here we'll be calling it as a small molecules. So in case of biologics, here we'll be talking about the protein model and everything. And here we'll be talking only about chemical compounds. So it will be useful for the chemistry people as well as the pharmaceutical people. And those who are in the pharmaceutical industry, it will be helpful for them. So if we come for bioinformatics, it will be helpful for most of the people who are there in the bioinformatics as well as cheminformatics approaches. And coming to bioinformatics, so there is something called target selection. So what does target represent here? So in case of bioinformatics, we can call it as a protein target, and here we'll be calling it as a chemical compound or drug molecule or any compound. So those are called as target here. And here homology modeling. So in case if the protein structure is not available, so what happens is like I have a protein molecule, and most probably you might have discovered a drug and you want to test that protein molecule. And in case if the protein structure is not available, what we will be doing, we'll be taking that protein structure and we'll be doing homology modeling, and you will predict the structure. So that's all. We will be discussing about homology modeling today. And later on, if you have any doubts in that drug discovery process, we can go for after the session, like we'll go for molecular docking. What are all the approaches or along with that? So, and apart from that, you can go for protein engineering. Protein engineering is nothing but we'll be studying about the mutation analysis in the protein. I have predicted my protein structure, and I'll be studying about the mutation which is present in the protein structure. So that's why we'll be using protein engineering approaches. Apart from that, genomics and genomics is nothing but if you have your entire genome, you want to study about the gene particular mutation, you'll be going for genomic studies. So probably you might have heard about the term personalized medicine and the NGS approaches. So NGS and personalized medicine, which comes under genomic study. So apart from that, protein developability here represent about the antibody complexes. And protein docking, you have one protein molecule and you want to see the interaction between two protein molecules, you'll be going for protein docking approaches. Apart from that, you will be something called immunogenicity. So what is immunogenicity here? You have your antibody molecule and you want to check that antibody molecule and how the antibody interacts with the particular compound. You can check through immunogenicity approaches. So these are the things are there in the bioinformatics. There are n number of fields are available. So here I'm representing only few of the terms which is familiar to the people. So apart from that, in cheminformatics approaches, so what we will be doing in cheminformatics approaches, I have lakhs and thousands of compounds and I want to screen that particular compound. So what I'll be doing is I'll be doing virtual screening approaches to perform it. And apart from that, there is something called structure and fragment based design. So I have my ligand molecule. I want to predict the particular structure as well as a particular fragment. I can replace that particular fragment through cheminformatics approaches, and then I can design the drug based on the fragment. So that's why it is called structure and fragment based design. And apart from that, there is something called pharmacophore modeling. So pharmacophore is nothing but you have your ligand molecule and you have certain features. So number of hydrogen bond donors, acceptors, so it will be there. So with that pharmacophore model, you can predict your structure, predict your ligand molecule. And apart from that, there is something called quantitative structure activity relationship. So if you know your IC50 value of the particular compound, you can design your own model using QSAR approaches. So, and apart from that, library design, you can design your own library as well as diversity and Pareto analysis. And apart from that, admit and toxicity. So in wet lab studies, you can study with cell lines, but here what happens is admit and toxicity studies, you can study through bioinformatics approaches. There will be set of parameters are available to test it, and with that, with the help of those parameters, you can conclude these are the specific toxicity or specific molecules. It can fall under this category. So that's why you will be doing admit and toxicity study. Apart from that, there is something called molecular dynamic simulation. So I guess most of them are aware of this molecular dynamic simulation. So what happens is you have predicted a chemical compound and you have some protein structure. So what happens is you want to go for docking. So you have done how the molecule has been interacted with a particular compound, and you find how the interaction has been happened and all. So finally, what happens is once the interaction has been done, so you'll be studying that protein ligand interaction in atomic level. So we are all very well know we are made up of atoms. So the proteins which we are studying, which is made up of atom molecules. So what happens is if your drugs has been interacted with your particular molecule, particular protein molecule, that protein molecule has atoms in that. So you can study the atomic level interaction through molecular dynamic simulation. So these are the approaches are there in the bioinformatics as well as cheminformatics approaches. So coming to computational method. So we'll be having that question. So we have bioinformatics as well as cheminformatics approaches. So what is computer-aided drug design? So computer-aided drug design can be categorized into two types. One is structure-based drug design and another one is ligand-based drug design. In structure-based drug design, there will be something called homology modeling. Homology modeling is nothing but you have your protein structure and you want to predict those protein structure. So you will be modeling your protein structure, and after that you will be performing molecular docking study and then molecular dynamic study. So this is one of the structure based drug design approach. And here in case of ligand based drug design, you will be having QSAR, pharmacophore, similarity searching as well as virtual screening. So these are the some of the methods which they are used for structure-based drug design as well as ligand-based drug design. So in case of chemistry people as well as pharmaceutical people, so what happens is you have synthesized your compound, you want to go for pharmacophore studies, you can go for similarity searching as well as virtual screening, and you can end up with a particular compound. So what happens is you will be ending up with a particular compound and you will be doing the interaction, and then you can go for structural studies. In structural studies, you will be performing molecular dynamic simulation. So this is the major difference between structure-based drug design and ligand-based drug design. So most of them have covered about the ligand-based drug design. So here today I'm going to discuss about, so you have already predicted your ligand compound and you want to study about the interaction between the protein as well as ligand. If the protein structure is not available in the homology, if it is not available in the RCSP protein data bank. So you are all aware of protein data bank. That is, a protein data bank is a repository which contains all the protein molecules. So whatever they have done X-ray crystallography and NMR spectroscopy study, they will be predicting, once they have predicted the structure, they will be depositing those structure into RCSP protein data bank. So once they have deposited that structure, in case if the structure is not available, what in case if the structure is not available, it will be performing homology modeling. So what are all the steps involved in structure deposition. I'll just give this, it's extra information. The reason why I'm giving this extra information, because most of them are not aware how the protein structure has been deposited. So it's purely bioinformatics approach. Say for example, they have studied a protein structure, and once they have studied the protein structure, they got the protein structure itself, and then they have to deposit in a particular repository that is called RCSB protein data bank. The default protein data bank is available. So what happens is how they will be depositing the protein structure. So first of all, they will be checking the sequences. So they'll be checking the sequences through BLAST-P. So you're all aware of BLAST as well as BLAST-N. BLAST-P is protein. You can study the sequence against a protein, and you can search for the sequence against nucleotide. So this is what there in the you have to check the sequence. And once you have checked the sequence, in case in your X-ray crystallography structure contains ligand molecule, you'll be checking those ligand, that is your chemical component dictionary. So for this also, you'll be having some separate like links are available in the RCSB data bank itself. So if you go through this link, you'll be having that option to explore how the ligand has been deposited and how the protein molecule, how the protein structure has been deposited into the protein data bank. So after that, so once you have checked the sequence, and after that you have to check your ligand molecule. So once you have done both the sequence as well as ligand molecule analysis, after that you will be going for, you have to prepare your data. You will be having some data, and you will be preparing those data for your deposition using your PDB extract as well as the SF tool. So these tools also are available in the RCSB data bank itself. And once you have prepared the data as well as once you have extracted the tool, and then you will be validating your structure. So once you have validated your structure, you need to deposit the structure through ADIT as well as ADIT NMR. In case if it is X-ray structure, you will go for ADIT, and in case if it is NMR structure, you need to go for ADIT NMR. So this is the major difference between ADIT as well as ADIT NMR, and these are the steps which is involved in structure deposition through RCSB. So coming to the protein structure prediction method. So today we will be discussing about homology modeling and how we are going to perform homology modeling using BioVia Discovery Studio. Say for example, there are some protein structure prediction methods are available. So we are all very well aware of that. In case if the protein, I have already mentioned here, through X-ray crystallography as well as NMR structure studies will be predicting the protein structure. What in case if the structure is not available? So there are 3 computational methods are available to rectify this problem. So say for example, there is something called ab initio method. If the protein structure sequence will be searching against the blast. So in case in the blast will not be getting any similar sequence, then we have to go for ab initio method. This is for starting from the scratch. You doesn't know anything about the structure. You doesn't have any similar structure, and you will be going for ab initio method. And apart from that, there is something called threading. And what happens in threading is you will be having 40 to 50% similar sequences will be available. So based on that, you can model your structure. And here you will be getting the protein similar 3D structure. You will be getting the RCSB PDB data bank structure with similar sequences. So based on that, we'll be performing homology modeling. So today we'll be discussing how the homology modeling has been performed using Discovery Studio. And before we start with Discovery Studio, like why do we need to go for this particular software? So there are n number of online softwares are available, n number of commercial resources are available. Why do I need to go for this particular software? Say for example, the force field. The force field completely differs from other software. So say for example, CHARM, and CHARM is one of the topmost force field which has been used for molecular dynamic simulation, and NAMD is also one of the topmost force field. And apart from that, protein homology modeling will be using Modeler. Modeler is the first developed algorithm to develop a protein molecule structure. So, I mean, we have some experimental method, X-ray crystallography, NMR structure, NMR prediction methods are available. Apart from that, in case of bioinformatics approaches, Modeler is the first algorithm which has been completely developed to design and develop a protein structure model. And after the model has been developed, then only they have started developing some, you might have heard about Swiss PDB and all. So after that only they have developed the Swiss PDB and those modeling structure, model structure databases and all. So apart from that, protein docking will be having, I'm sorry. So for protein docking, we'll be having ZDock algorithm. Apart from that, we'll be having for protein aggregation and viscosity, we'll check with ADMAP and SCM. So these are the well-versed algorithms for to developing your protein aggregation as well as viscosity. And apart from that, there are many more novel as well as internally developed peer-reviewed scientific algorithm has been embedded into Discovery Studio. So that is a major difference over other commercial software. And before we start with that, you should know what is homology modeling, and it's a basic steps about homology modeling. Say for example, you have two homologous protein. Homologous is nothing but you will be having two similar proteins, and you need to check for the conserved region. I mean, if you check for the portions, it will be similar. So you have a sequence A and sequence B. Both the sequence contain similar sequences. That is called portion of two protein with similar structure, and those similar proteins will be highly similar proteins. Those are called homologous protein. And what happens in homology modeling is like you will be having comparative modeling. So what happens in comparative modeling is nothing but you have two similar sequences, and you will be comparing with the two similar sequences. After that, you will be predicting the protein structure, which is based on the template. One second. Based on the I'm sorry. So you'll be having two structures. Say, for example, you have two protein structures and you will be comparing those protein structures. That is called comparative modeling. So apart from that, you will be having called predict protein structure, which is based on the template. You'll be having, you will have to predict your protein structure based on the template molecule. What is template here? I have some sequence and I need to go for that. So this is my sequence. I need to check for the similar sequences. That is called template. And after that, analysis of protein function. So what happens in homology modeling is you'll be analyzing your protein function as well as interaction and its antigenic behavior of the protein, and you can rational design of your protein with increased stability as well as the novel function. And if you see, if you get 30% of the sequence identity that is required to complete the homology modeling. So whatever I have mentioned, so this is called sequence. You will be having your protein sequence. You know very well about the cell and the central dogma of the cell. And in that, from the central dogma of the cell, you will be having DNA has been converted into RNA and RNA has been converted into protein. So through translation, we have got the protein sequences. Those protein sequence only here we have mentioned. So if you have this protein sequence and you have to find your template sequence. What is template sequence? You need to search for this template sequence through RCSB or any other database. And if you find the template sequence, and you can easily model your structure. This is your performed homology modeling. And here template sequence. So this is your target sequence. And once you have get your target sequence, then you will be predicting your protein structure. So after that, if you go for so why homology modeling is more important? It is completely based on the evolution. The structure is more stable, and the changes occurred at the slower rate when compared to its sequences. And you can see the distantly related sequences fold into similar structures. And as well as 40% of the homology or similarity are guaranteed to adapt a similar structure. So this is the basic understanding of homology modeling. And so what are all the steps involved in homology modeling? So how we can perform homology modeling? We'll be having certain steps. Say for example, you have to compare the sequence and you will be identify the closely related sequences. And once you have aligned the closely related sequences, you need to align the sequences with the template. So say for example, you just take this example as a COVID sample, and you will be having your target sequence as your COVID sample, and you need to identify the template identification. SARS or miss. So you will be you have to check your SARS or miss sequence, and once you have checked your sequence, you have found one template identification, and once you have identified this particular template sequence, then you are going for alignment. So alignment is nothing but you will be having similar sequences, and this alignment can be based on structural alignment as well as multiple sequence alignment. So once you have aligned your structure, I mean, based on the structural alignment is nothing but you might have studied about, you might have know about protein structures, I mean, alpha and beta helix and all. So it will be based on the structural alignment, you will align your structure, or else you can go for multiple sequence alignment. And then so once this alignment has been done, you can build your model. So, and once you have built the model, you can refine your model and you can evaluate your model. If it is good model, you can go for it and use that model for particular approaches. So you have synthesized your compound, you want to go interact, you want to see the interaction between your protein as well as your compound, you can use this particular model. In case if the model is not good, what you have to do is you need to check whether the alignment is problem or whether the template identification or selection is the problem. You need to understand what is the exact error in this. So after that, if you rectify it, you can get the good model. So once you have done the model, how will you evaluate the model? So obviously through Ramachandran plot, you will be evaluating your particular model. So these are the steps, basic steps involved in homology modeling. So we'll be having sequence alignment and template recognition, as well as model building, alignment correction, and backbone generation. So backbone generation is nothing but you have your compound and you have your protein molecule, and your protein molecule has backbone. And if the backbone molecule is not modeled properly, you can generate it based on that. And then loops or variable region. In case if you have modeled your protein structure, and there is loops will be available, the protein loops will be available. If you want to rectify the loops designing, you can go through loops or variable region modeling in the same protocol. And then side chain modeling, in case if the side chains are missing in the protein molecule, you can go for it. And then optimization, you can optimize your molecule, then further which is followed by structural refinement. So once you have predicted your structure, you can go for structural refinement. How will be doing structural refinement? So you have your protein structure and you'll be predicting through Ramachandran plot, or there are n number of servers are available. So whatever I have mentioned here, these things can be done through online servers also. Say for example, Swiss PDB, and you can try with modular software also. The modular software is available. If you go and check in the modular website, you will be having that exe file. Once you have downloaded your exe file, you can perform the same steps. So it will be asking for your target sequence as well as a template sequence. So you can perform modeler only if you have template sequence. In case if the template sequence is not available, then you cannot perform through modeler, and you can try with Swiss model. There is something called Swiss model website is available. You can try with that. Apart from that, Robusta, ITaser. So these are the, for ITaser is for threading related modeling structure. And like that, you'll be having many homology modeling related servers are available in the online. You can try with that also. So homology modeling with modeler. So I'm just explaining the algorithm which is present in the modeler. So say for example, you will be having automated homology modeling program, and here you need not assign the manual assignment of coordinates. So it has been developed by Dr. Andrew Sally of the University of California, and the main aim of the modeler is to predict the probable structure. You have a structure and you have your sequence, and this sequence, based on the sequence, say for example, so here this is your sequence, and based on this sequence, so you have some target structure. Based on the based on BLAST, you have got target structure, and you are identifying your structure based on the main specific target sequence. That's why modeler has been used, and you will be predicting the same homologous structure based on the template sequence. And apart from that, it derives a 3D model which is completely close to the template structure. Also, it can satisfy stereochemical as well as homology derived spatial restraints. And what is the exact modeler workflow? So you'll be having an input as well as the alignment between the target sequence as well as template sequence. So what happens is you can derive a set of restraints, and here you will be having homology derived restraint, and this can be calculated, which is based on the template. You can calculate the distance as well as dihedral angles here. And coming to stereochemical restraint, you need to check for the bond length as well as the valence bond length, bond angles. And apart from that, there is something called derived probability density functions. Based on this function only, it can be predict the complete geometric features of your protein structure. And once you have predicted it, and you can create the objective function from the molecular PDF that is also combine all the features PDFs. And so once you have predicted this, then you will be having that objective function in Cartesian space. So this is the exact workflow which has been used in the modeler algorithm. So if you need to study more about this, you can go to modeler website, and you will be having some PDFs are available to how the modeler has been worked, and what are all the restraints are available, what is probability density function score, and why the probability density function score are more important, why it has been used, why it has been calculated the geometric features. So these things will be available in the modeler website itself. So coming to sequence searching for homologous protein. So what happens is so this is completely from the BioVia Discovery Studio. So whatever I have mentioned here, these are all you can do in the online servers. And here what I am representing, so whatever I am representing, are you audible? Am I audible?
Yes, ma'am.
Yeah, I'm sorry, I lost my connection actually.
No problem. Yeah. Continue.
Okay. So what happens is you need to seek yeah. So what happens is you need to search for your sequence. So based on the homology modeling. So whatever I have mentioned in the previous slide, till the previous slide, it has been taken from, you can do from online servers as well as online platforms. So whatever I'm showing it here, it can be done only through BioVia Discovery Studio software. Say for example, how we'll be doing homology modeling using this particular software. You'll be having sequence searches for homologous protein. So I have already mentioned you have to search for the similar sequences. So here itself you can see, here you'll be having your particular sequence, and once you have taken your sequence, and you need to search for other sequences. So once you have got the sequences, you need to check for the template. So you'll be aligning with the template, and these are the similar sequences. Say for example, in the other softwares and all, you need to search for BLAST. So first thing is you need to search for your target sequences through BLAST, and then template sequence through BLAST. Once you have got the sequences or your protein structure, then what happens is then you will be performing your you will be, once you have performed your BLAST search, then you will be having identified sequences. So here what happens is the BLAST has been embedded into the particular software. So here you will be having your sequence and your template sequence. And once you have got your template sequence through this, say for example, this is your sequence, and this is your target sequence, and here you will be having your template sequences. And 1 second. So you'll be having your template sequences. So you'll be having 15 template sequences are available. So what happens is you'll be having the same sequence length, alignment length, as well as the bit score, as well as the E value and positive, as well as resolution. So like that, based on this criteria, you can filter your molecule. So once you have done your sequence analysis, what happens is you can predict this particular, you can take this particular protein. So say for example, this protein structure is completely 44% identical. So what happens, we will be taking this particular structure and we can go for the further steps. Say for example, the next step for homology modeling is you have to assess your template. So how will you assess your templates? So you will be assessing your template based on your so you'll be assessing, yeah. So you'll be assessing your templates based on the assessment similarity as well as multiple templates. So first thing is you have your protein structure and you got your template structure. So once you have got your template structure, what happens is you need to go for alignment. So you'll be aligning your compound and then you'll go for the multiple sequence alignment or structure based alignment. So if you got similar structure based on the structure alignment or multiple sequence alignment, then you will be predicting your model. So this is how the assessment of template works. And once you have opened your template sequence, you can see the identical. So say for example, you can see, so here the arrow mark represent the alpha helix as well as beta pleated sheets and all. So you can see those criteria, homologous region, and you can finalize. So this protein you will be getting the 44% identity, and based on that identity, you can predict your model. So this is how the assessment of template works. And apart from that, you need to, the third step in homology modeling is you need to align your target sequence based on the template. So you'll be aligning your sequence to the template and you'll be validating the alignment. So say for example, you have your sequence and you have to check for the template. So you have already got your template structure here. So once you have got the template structure, you will be aligning your sequence to the particular template. Once you have aligned your structure, you have to validate the alignment. So once you have to validate the alignment, you will be building your model through homology model building. So and once you have done the modeler, through modeler, you will be building that model. So after that, you can go for model analysis as well as selection. So once you have got this, like you will be going for analysis of PDF energy. So you have got your homology modeled structure. So once you modeled your structure, then you will be analyzing your PDF energy, that is your probability density function score as well as dope score. So once you got the PDF score as well as dope score, you will be analyzing the model based on the template molecule. So and how will you doing this? So say for example, you have two structure and you need to superimpose both the structure. So once you have superimposed both the structures, then you can go for analyzing of the template molecules, and then model refinement can be done through this. So coming to the model refinement, you will be having refinement will be available. Apart from loop refinement, you can do side chain refinement as well as the constraint minimization as well as the dynamics. Constraint minimization and dynamics could be done through this modeling structure itself, and then you will be performing. So what happens is I'll be summarizing the protein model. Say for example, you have your sequence structure and you need to go for alignment. So once you have aligned your structure, then you have predicted your homology model. So once you have modeled your structure, then you will go for model refinement. So how will you choose the best model? Will be question from the beginning of the presentation. I have started with template sequence, target sequence. So I'm just representing again the template here. Mention target. Your target sequence is your sequence which you want to find out the structure, and template sequence which represent the structure which is going to be a similar structure, similar homologous structure. So once you have got the structure, then what happens is you will be comparing both the structures. You'll be comparing your target sequence with the template sequence, and you will be identifying the homologous properties. Once the homologous properties has been identified, if the protein similarity has been identified, then we'll be performing then we'll be performing the alignment. And once the alignment has been done, then you are building your own model. So how will you choose the best model? You will be choosing your best model with the lowest score is completely based on dope score, as well as PDF total energy, as well as PDF physical energy will be there. So what you will be considered as the first energy? Say for example, first you have to consider your PDF total energy. PDF is nothing but probability density function score. So once you have taken your PDF total energy, then you have to look for the dope score, and at last finally you will be finding out the physical energy. So how will you choose the physical energy? So you will be having an option for PDF total energy dope score, and lastly the PDF physical energy. And then the second step is you need to verify your protein. So you need to verify your protein through profiles 3D, and you will be calculating whether your structure is compatible with its sequence, and you can also calculate the compatibility of each residue. You will be having your sequence with its predicted 3D environment, and once you have calculated the compatibility of your each residues, and here each residue is mentioned about your sequence contains amino acid residues, and those amino acid residues compatibility you can look for the compatibility between the sequences which has been predicted in the 3D modeling. So once you have reduces the 3D structure, you need to look for the 3D structure in the 1D string, and you need to check for the residue environments too. And after that, once you have calculated the verify score for template as well as your model, you need to look for the score between the template as well as your target sequence. Your model, the sequence. So once you have taken your model sequence as well as your template sequence, you need to look for the multiple structures as an input, and you will be checking how the protein has been differed from this model protein. So these things you need to look for it. Apart from that, you need to close the calculated verify score, and it has to be expected high score and also the better model. And once you have modeled the verify score and at the lower level, and the expected low score, it will be poor. So apart from this, so you have calculated your dope score, PDF total energy, and PDF physical energy, and you are not satisfied with the result. So what happens? You are going for run verify 3D. So here you will be checking your PDF protein structure, and you will be comparing your template structure. And once you have done with your profiles 3D, then you have to obviously you need to go for Ramachandran plot to identify the outliers of the model which has been designed for your work. So this is how you need to select your best model. So coming to identify the poorly modeled regions. So what happens is, how will you identify the Ramachandran plot result? You'll be identify the outlier residues in the model, and you need to align it based on the template and also based on the display in one plot. If so, they might not be concerned, and if not, possibly the misfolded region occurs. And even if you have symbols like a glycine as well as a glycine as a triangle, and prolines are square, and also other types of circles are available in the identified poorly modeled regions. So this is what there in the homology modeling. Until that, if you have any question, you can ask me, and then we'll. Till that, if you have any question, you can ask me.
Any questions from the audience? You have any queries? You may text in the chat box also.
I'll show you another ligand-based drug design method. So till that if you have any question till homology modeling, you can raise the question. Since it is new to pharmaceutical people, This is the main. So you have designed your drug. Once you have designed your drug, obviously you will be checking with cell lines. So after checking with cell lines, what are the other sources are available? If you look for the computational approach, obviously this is say for example, you need to go for homology modeling, and you will be checking your structure in case if the structure is not available. So in case if you have your structure with your in the CSV PDB data bank, you can directly download your structure and you can go for it. So I'll share this PPT.
Any queries? Please raise your hand.